This is our third set of evolution notes under Big Idea 1, and it deals with phylogenies. Okay, so what would you say this organism is? Is it a snake? Is it a lizard? Maybe upon first look you would say snake, like I would instinctively say. It's got that characteristic snake-like body, no limbs, looks kind of just like a snake. But this is actually called the legless eastern glass lizard. Okay. So how would a scientist go about determining what this actually is and how it fits into our evolutionary history? What groups is it more closely related to, etc., etc.? So we can do this by looking at or comparing traits. One way is to do that through DNA, now that we have the technology. Another way is just to look at some of the features of the organism. So if we take a look at this eastern glass lizard, we know that it does not have a highly mobile jaw, so it can't open it as wide and move it around like a snake can. It doesn't, despite its appearance, have a large number of vertebrae like snakes do. Nor does it have a short tail located behind the anus. So snakes actually have a very short tail behind the anus. Most of the body is, all of the organs and everything are elongated in a snake. And then there's the anus and then a very short tail. The eastern glass lizard actually has a longer tail after its anus. So when we study these, we're studying evolutionary histories or phylogenies, which is of a species or could be a group of species, and we're looking at how they could be related to each other. We use a process called systematics that's going to help us reconstruct and interpret an evolutionary history by trying to look at relationships or evolutionary relationships. Before we get a little bit further into that, we need to talk about taxonomy. I don't know if you remember any of this from your regular bio classes, but in one of our first sets of notes, we talked about Linnaeus, and we said that he was a biologist, Carolus Linnaeus, who came up with our two-part naming system that we still use today, and that is the binomial nomenclature. Okay? So we're going to talk a little bit more specific about that. But before we do that, let's just refresh our memory on why we can't use common names, or why common names can be confusing for scientists to use. When you say the word monkey, many of you get an image of maybe something that's not even a monkey, like an orangutan. Or maybe some of you see a capuchin monkey. Just so you know, there's all different kinds of monkeys. Many things happen with trees, like we will call a lot of things pine trees, and they're not really even a pine tree. They might be a spruce or a fir. We just say, oh, it produces a cone, so it's a pine tree. Okay, so we have that where one name could refer to many different organisms. Or we have times when our names don't really make sense for organisms, like jellyfish, crayfish, and silverfish, none of which are actually fish at all. So in Linnaeus' system, there's two names. They're both Latin. The first part of the name is the genus, and that is a grouping to which the species, which is the second part of the name, belongs. So species is the most specific that you can get. We call that the specific epithet, and it is unique for each species within that genus. Canis familiaris are dogs. Now, breeds are another thing. When we talk about breeds, there are many different kinds of breeds of dogs. That's like another subdivision, or we could maybe even call it a subspecies. But all dogs are Canis familiaris. But they belong to the genus Canis that would include things like Canis lupus or the wolf. Okay, so you have the two-part name. It's Latin. The genus is the first, species is the second, and genus is capitalized. Another thing that you'll note, I actually put genus and species in italics because that's how they would be presented in typed form. So if you decide that you want to name an insect that you discover after a friend, you can do that. But one of the rules of the naming council is that you need to make it Latin by adding like a US or AE or Latin sounding ending to the end of the title that you give this organism. Okay, so we're going to review a little bit here that hierarchical classification. So in the first chapter, we talked about atoms to molecules. Molecules make up cells, or make up organelles, which make up cells, which make up tissues, etc., etc. Now we're talking about organization on a different sort of level. We're putting organisms together based on similarities that they may share. And you could look at this as we're getting more exclusive if we go from domain to species, or what most scientists say is increasing inclusiveness when we go from species all the way up to domain. Okay, so species that are closely related are going to be grouped into the same genus. Genre, which is plural of genus, that are closely related will be grouped into the same families. Okay, families to orders, orders to classes, similar classes to phyla, similar phyla to kingdom, and then finally similar kingdoms to domains. And remember, we have those three domains: eukarya, bacteria, and archaea. Each level, domain, kingdom, phylum, class, order, family, genus, species, is called a taxon or taxa for plural. Okay, so we're going to look at it in examples so that you can kind of see how all of this works, refresh your memory. Okay, so if we're going from the species, the specific, to the more general, we have the Panthera pantris, that's a pretty blurry picture, but it's a panther. Okay. All of the Panthera will be included in this genus. So there's going to be other like organisms, try and change the color there, that are within the same genus. That did not work. Try that. Okay, there we go. That are in the same genus. Then you're going to have several genuses, so there might be Panthera and maybe something else, I don't know, of the lineage of this panther, but that would fit under the family Phylogeny. And then the Phylogeny are going to be in with the canines and some other organisms, and they're going to fit under the order Carnivora. Okay. And now we're going to get even more inclusive and say, well, now we're not just talking about carnivores, we're also going to talk about herbivores and omnivores and pretty much anything that gives birth to live young and has mammary glands. So we'll put it in the class Mammalia. That's not inclusive enough, so then we'll go up to phylum, where we're saying now we're looking at anything that has a spinal cord, a chordate. So vertebrata is actually a subphylum, so it's not its own phylum, it would be a subphylum. So anything that has a spinal cord would be in the phylum Chordata. And then finally, we could include all of the animals. So we've got the invertebrates and the vertebrates that would be in the kingdom Animalia. I guess that wasn't finally, because there's one last one, and that's the domain. And so finally, in the domain Eukarya, you've got not only the animals, but the plants, the fungus, and the protist. So very distantly, we as humans are related to fungus. Okay. So Linnaean's system does not necessarily reflect an evolutionary descent. It can, but that doesn't mean that it necessarily does. Phylogenetic trees, branching diagrams, evolutionary histories, cladograms are a little bit more specific into what the evolutionary history could have been. Sometimes through Linnaeus' system, we'll get an organism that's classified incorrectly because we've uncovered new DNA evidence. That doesn't always happen. Most of the time, he was spot on. Most of the organisms fit where Linnaeus had them. But occasionally, like I said, that happens where an organism, when we uncover DNA evidence, we'll find doesn't fit with where we had it before. Okay. And that's okay, because they can reclassify it, put it in a different area, maybe change its genus, change its order, whatever it is that we need to. They can go through the naming council and go ahead and change anything that needs to be changed based on DNA evidence. So if we take a look at an example of a phylogeny, it's a little bit different than the clades that we may have looked at in class, but in some ways very similar. Okay, now one thing that I want you to remember is phylogenies are hypotheses about evolutionary relationships. So it could be your hypothesis, it could be my hypothesis, it could be some other scientist's hypothesis. It's a good guess as to what we think happened, and many times they're backed up by much evidence. But again, they're just hypotheses. We cannot say 100% this is exactly what happened. Okay. Usually when we have a phylogeny, we're going to depict it as a series of what we call dichotomies or branching points. Okay, so like there's a branching point. Here here's a branching point. Is it going to go here? Is it going to go here? Okay. Each branching point is going to represent the divergence of two lineages from each other, from that common ancestor. So back here is the common ancestor, and then the branch indicates two pathways that the organisms could have taken from there. We have over here things that are called sister taxa, because as you can see, if we follow our diagram, these are closely related to each other. And so we're going to call them sister taxa. Sister taxa work on rotating branches. So you could actually flip this, and this could be CB. So it doesn't really make a difference. Now what you can't do is flip A and B, or A and C, but you can flip CB with each other. Okay, I hope that makes sense. And then we can say they're rooted in the most common ancestor, or most recent common ancestor. So everywhere, so if we're looking for where B and C are rooted, it would be in their most recent common ancestor. Okay, so there are some limits to phylogenetic trees that we should discuss. One thing is that we can't assume that one taxon evolved from the next one. So we have our little branching diagram here with the sister taxa. We can't say that B evolved from C or C evolved from B, but we can say that they did have a common ancestor at some point in time. Okay. We can't also say that this sequence necessarily indicates a specific amount of time. Like right here, we can't say, oh, that's 1,000,000 years. We have to be told that. If the person who's setting up the phylogeny or the branching diagram says, okay, each 5 centimeters or 3 centimeters is equal to 40 million years, then that's what we know. But unless we're told, then we can't assume that. And the other thing that we have to remember is phylogenies are intended to show patterns of descent, not what looks like each other. Okay, so crocodiles would be more closely related to birds based on their similarities in molecular similarities and some of that bone and skeletal similarities, but they are still grouped with the reptiles if we're looking in Linnaeus' system. Okay, so how do we apply, or how do scientists apply phylogenies? If we can discover who the relatives of certain species are based on constructing these phylogenies, we can actually use those relatives as a potential genetic reservoir for alleles to be put back into a population in case of disease or some other catastrophic event. So an example of that is maize or corn. Very long time ago, corn used to be called teosinte, and it was actually not very appetizing at all. Very small, hard kernels that we wouldn't have wanted to eat. We bred and bred and bred and bred, and now we have the big, beautiful, juicy ears of corn that most of us like to eat in the summer. Okay, but that's not its natural state. If something happens to that corn, we can actually go back to its closest relatives and try to breed new genes, or actually old genes, back into the corn, our new corn, to get it to maybe even be more cold resistant or disease resistant. So that's one way we could use a phylogeny. Another way is we can actually do some detective work and infer species identity by analyzing how close the DNA is related based on different organisms. So if, excuse me, somebody has some meat and they say it's whale meat, but they're swearing that it's not one of the endangered ones, you could actually test that, find out what the DNA evidence shows us based on a molecular basis, and we could look at the phylogeny and say, well, this whale and this whale share these similar genes, so it must be this endangered whale here. And so they can use that to, just keep track of organisms, to identify organisms, to find out if people are doing things that they shouldn't be doing, like poaching. Now we have to learn how do we actually... Okay, so how do we construct these? What are the first steps? The systematists, which are the scientists who do this, first have to gather as much information as they possibly can about the organisms, morphology, genes, biochemistry, anything they can. The general rule is that if two organisms share similar morphology, they're probably closely related, but that isn't always the case. An example of where we can get confused, where we think that the morphologies are different, but they're actually closely related, are the silver sword plants in Hawaii. Silver sword plants can either be tall and very twiggy, or they can be dense and ground hugging. So despite that phenotypic, the phenotypic difference, the genes are actually very similar in those two organisms, those two types of silver sword plants. So we would say that those are closely related, even though their morphologies aren't exactly the same. We also need to be careful of confusing analogies, and not English analogies, analogous characteristics. We talked about those in the first set of notes, that can be formed through convergent evolution. We want to make sure that we're not putting bats and birds together just because of the fact that they have wings, when in actuality, if we take a look at the difference between a bat and a bird, we can see more similarities between a bat and a cat than a bird and a cat, or a bird and a bat. Did I get all that? Hopefully that made sense. So we want to be careful of those. Okay. Then we can also look at that DNA that we've been talking about and comparing DNA. DNA comparisons can be very tricky. Thankfully, we have computer programs that help us estimate the best ways to align the genes. And so we can rely on our computer programs to do this instead of trying to go through the painstaking work of looking at hundreds, even thousands of base pairs. Okay. So one thing that we have to remember is that very closely related species may only have one or a few sites that are different. Like it might be, two A's that are different, or ATG on one and AAG in another. That makes a big change in the organism. Another thing that can happen is sometimes genes are deleted. There's different kinds of mutations, which we'll talk about later. One of those is a deletion, which means we've actually lost part of the gene, or only one base, like one letter, A, T, G, or C, on that gene. When that happens, if you remember from biology, it shifts everything over one. So if you're looking at those two sequences, you may not think that they're like similar at all, but the computer program is able to pick up on the fact that, if we shift everything over one, then now these line up perfectly. So we can see where a deletion occurred and where two organisms may be closely related because of that. So here's an example of that. We have our DNA up here. We're going to take out this one little bitty letter, okay? And that's going to change everything. Here, we're going to insert GTA. Some mutation comes up, something happens, and we get a part of a gene inserted, or something could be even replicated. So our change from the top line here to right here is huge, okay? Even though we've only taken out one letter, it's made a big difference, okay? And here, we've actually added a sequence right in there, okay? So now that's made another change. And so when we're trying to compare them, we'll pull this up a little bit, we might think that they're not closely related, but they actually are. So the computer could line up, oh yeah, they both start with this CCAT, and then there's some blanks here, but then they match up again here, and then they match up again here. So, the computer can help us determine, there was a deletion here, and there's something extra that's going on here. So hopefully that makes sense as a start to how we construct these. Next time we'll be talking a little bit further on actually putting them together, making charts and tables to fit them all together, and we'll practice a lot with these things that are called phylogenies and making these evolutionary histories. See you later.